as that's true. In the summer session, we have so little time. So, as you know, you have an anatomy test on Monday. You then have, which I think it's the 20th, is the only Monday you don't have a test. And then following that, you have both an anatomy and a physiology on that Monday. So be prepared for that. Be psychologically prepared. You can have that's going to come fast. Also, we're going to have to make a mild scheduling adjustment because of whatever reason. We have, I think, too many anatomy, and we need to flip-flop those with physiology at some point. I don't want to burden you at the end of the course with doing that. So at some point, very shortly, we're going to swap one of the anatomy and physiology sessions. As I said, in one of your sessions, you only have eight hours of anatomy to be tested on. And I'll look at the schedule, and as I emerge from my illness, I'll make sure that I attend to the details to make sure that everything's properly balanced. But again, I guess the major thrust here is those of you who are not doing well, and you may want to face that at least at this point on the first test, please take the corrective remediative action, because it just comes so rapidly, and it proceeds and unfolds so rapidly that we don't have time here to do a lot of catch up. So again, I'll be at the office if you need me, or get with somebody. You have a lot of folks in here that are doing very well, and get with them if you need to for assistance with the course material. Okay, so we want to conclude our discussion of muscle. By the way, before we move on to smooth muscle, I'm going to abbreviate this unit dramatically relative to what's in your slides, so we're going to go through this very quickly. But I noticed in my effort to summarize the material last time on skeletal muscle, I overlooked a couple of things. So please add the following. You probably already know this, but add the following point, because I want to make sure you understand that those three things that we said are important for skeletal muscle contraction: calcium, ATP, and magnesium. They all have to be available. They all have to be readily available, and they all have to be limited when the muscle enters relaxation. Right. So if there's insufficient calcium, that means the contraction cannot occur. That means the cross bridge cycling and all that cannot occur. If there's insufficient ATP, and this is the point that I wanted to mention to make sure you understand this term, if there's insufficient ATP to metabolically sustain the cross-bridge cycling, the muscle goes into what is called contracture, not contraction, but contracture. Contracture, and so contracture implies that in effect, by virtue of the insufficiency of ATP, the muscle is more or less in tetanus. Contracture is a special species of tetanus in which, the best example of contracture would be rigor mortis, when the muscle shortly following death becomes locked or rigid, at least for a time, usually 24, 48 hours or so. And that's primarily because of the deficiency of ATP production. And then the tissues, the cells go off with lysis, they begin breaking down, and enzymes and so on, to what they can for a time, and then degradation and so on. So that process only lasts for a short period of time. The last thing I would say about contracture, though, is that this is, like so many of the terms we've talked about, it's a term that has multiple meanings. So the term contracture can refer to, for example, what burn patients have to deal with in trying to regain musculoskeletal function following severe scarring. There are types of genetic contracture, which over time, for example, Dupuytren's contracture, the first two digits, usually about the late teens, early 20s, will tend to come together, and the individual has a more or less permanently somewhat apposed hand, and usually that's corrected surgically. So just be aware that the term contracture has many meanings, but in our discussion of skeletal muscle physiology, the implication is that there's an insufficiency of ATP, and at least for a time, for a variety of reasons, for a time, the muscle is in a persistent state of tetanus.
Okay.
Yes?
Is that just because the ATPs needed to get the myosin head to unlock, basically?
Yeah, to detach. To detach, exactly. All right, as I say, I just want to make a few general points about smooth muscle. There's a little table here. Unfortunately, we don't have all the information we need to address each of these points. But you'll know at the top, you'll notice at the top there's skeletal muscle. Then the two types of smooth muscle we're going to talk about. I do want you to know those two types, and we'll talk about that in more detail in just a moment. And then there's cardiac. So be sure you know what we mean by neurogenic. Remember, neurogenic implies that the muscle can only contract in response to some type of action potential, neural signal. So we said the skeletal muscle is neurogenic. But notice that smooth muscle can go either way. Smooth muscle really depends upon where it is. It's sort of a tissue specific. Now, just because a tissue, you can see that the cardiac muscle is not neurogenic, that single unit smooth muscle is not neurogenic. Just because a tissue doesn't require neural innervation, that doesn't mean that it doesn't receive neural innervation. So make sure you understand that even in the heart and smooth muscle, although these types of muscle, we can take your GI tract out of your body. This is what Starling and Bayliss did in the 19th century, early 19th century. Take it out of the body of the dog, and it will continue to contract. You put a little Ringer solution, a proper saline solution. Even outside of the body, that smooth muscle in the GI tract will continue to contract. That's because it's myogenic. But I want to make sure you appreciate it. Nevertheless, there is extensive neural regulation of the enteric musculature, but in a number of ways we'll talk about later, but chiefly through the autonomic nervous system. That's something called the enteric nervous system. All right. Let's come back to pacemaker. Notice the striated, the skeletal muscle is striated, cardiac muscle is striated, smooth muscle is not. So smooth muscle does not have the banding. Skeletal muscle is voluntary, the other types of muscle are not. And by that means you have no volitional control over them. I don't want to talk about the point made in this table, but I do want you to be familiar with this term, syncytium. Did we talk about that before? Syncytium. Seems like that came up earlier, but in any case, by syncytium, CYT implies what? Yeah, cell, synergy, togetherness. Yeah. So a syncytium is a group acting as one, like all of you. You're a syncytium. And there are a lot of tissues, and various physiologists and anatomists argue and quibble about whether one tissue is a true syncytium or not. For example, the heart is your classic example of a syncytium because through a variety of mechanisms, it more or less contracts as a unit. We'll see that's not quite true. It more or less contracts as a unit. And then, as I say, some multi-unit smooth muscle, probably not. Single unit, well, we could argue. But in any case, I just want you to know the definition of the term. And then let's not worry about this. I'm going to come back to some of this later. The only other term I want to refer to or direct your attention to is the notion of the pacemaker. And a pacemaker simply implies that there is, let me reword this, that the muscle cell has the ability to depolarize on its own. Remember, we talked about depolarization with respect to neurons, nerve cells. And we've also said that the excitable tissues, at least to the point that we've talked about thus far, are muscle cells and neural cells. And what they have in common is they're capable of membrane depolarization. So the excitable tissues are capable of membrane depolarization. We've said that skeletal muscle depolarizes, the membrane depolarizes in response to a neural signal at the alpha motor neuron at the neuromuscular junction. But here's the new idea. And we won't explore it in great detail now, but I want you to be familiar with it because it's going to come up later when we talk about the heart. Some cells have the ability to generate their own membrane depolarization. And they can do that in a variety of ways. But one way of thinking about this is they're just sort of, from moment to moment, changing, they're just sort of vacillating or changing their membrane potential. And so they're constantly undergoing depolarization, and they get out of that, they repolarize, and then they go depolarize and repolarize. So by a pacemaker potential, it can originate in a variety of ways. We're just talking about the intrinsic ability of that cell to undergo depolarization and repolarization. And as you can see, smooth muscle, at least some types of smooth muscle, have the ability to do this. I talked about your GI tract smooth muscle. It has this ability, although the way it does it's a little bit unique. All right, so over on the right here, we're looking at some GI tract smooth muscle. Later, we'll talk about the organization of this muscle. But in most of your GI tract, all the way from your esophagus on down to your butt, you have a lumen, as you know, and there are various strata, there are various tissue layers in that lumen, or surrounding that lumen. But at the outer margins of the GI tract tube are two layers of smooth muscle. For the most part, there's some exceptions we'll talk about later, and they're arranged in different ways. So if this is the lumen of the GI tract, you're going to have one layer of smooth muscle that sort of runs around the GI tract lumen in this way, and then you have exterior to that another layer that runs along this pathway, and that's what we're looking at here. We're looking at the longitudinal smooth muscle, the smooth muscle that runs parallel to the axis of the GI tract and the intestines. And then at the top there, you're looking down the transections of individual smooth muscle cells that are arranged radially and like belts around the GI tract. But of course, when you think about the location of smooth muscle, the first thing probably you should think about is arterial smooth muscle, because arterial smooth muscle is responsible for regulating peripheral blood flow. And remember that too, to a large extent, is done homeostatically through primarily activity of the autonomic nervous system, the sympathetic division, but to a lesser extent through paracrine responses, local responses we will describe in more detail a little later. Unlike the very long cylindrical skeletal muscle cells, smooth muscle cells are rather small, mononucleate, and shaped like a porpoise. They have a fusiform shape. Another big difference between smooth muscle and skeletal muscle is, in addition to the fact that the cells are not long and cylindrical, rather they're sort of short and squat, they are not arrayed alongside each other like your desks here. Your desks are one row after another parallel. Not so smooth muscle. The desks would all be haphazardly arranged in various configurations because in smooth muscle, there's absolutely no regularity to the arrangement of the fibers. Here we're looking at some smooth muscle in a bronchial tissue. I just want you to sort of be aware of what you would see in a microscope. So this would be the epithelium out here. This would be what we would call the mucosa to the left out here. So all of that is a mucosa in here. And then this region out here is the smooth muscle. That's the muscle you see there. And so it's kind of wispy looking. You should be able to recognize this. What's that? Yeah, that's cartilage. That's probably in the bronchiole. Exactly right. That would be chiefly hyaline cartilage. And as I mentioned, for a PA, I think the first thing you want to think about with respect to smooth muscle is the arterial tunica media. Remember, the tunica media in both arteries and veins is the primary layer that contains the regulatory smooth muscle. And then once again, we're looking at the smooth muscle layers that make up what is called the muscularis externa, or the tunica externa in the GI tract. That would be down here. Then we'll have a submucosa. And then in the intestine, we have the little villi we'll talk about later, but all these little things up here are villi that help to facilitate the secretion and absorption of the hamburger that you had for breakfast this morning. Functionally, there are some important differences, and we're just going to summarize these between smooth muscle and skeletal muscle. First of all, smooth muscle is much more efficient. It uses less energy to perform more work, so it's more efficient. but it's less capable of powerful, rapid contractions. So it does not, smooth muscle is not capable of short, powerful contractions. Rather, smooth muscle contractions are relatively weak, but prolonged. It just goes on and on without fatiguing. And you can see some of that in this graph here. We have time of contraction over force of contraction. The magnitude is incorrect, but what this is showing you is that skeletal muscle contractions can be very, very quick, very, very rapid. Cardiac muscle sort of in between, but goodness, look at the smooth muscle. In fact, when we talk about the number of contractions, or we try to describe the rate at which smooth muscle contractions occur, for most regions of the body, we talk about the unit is not seconds, the unit is minutes. So we'll say that, for example, in the GI tract, there will be 10 to 15 contractions per minute. So that gives you some idea of the oscillatory time scale of these contractions and relaxations. There's a much higher concentration or much higher relative abundance of actin in smooth muscle than in skeletal muscle. You should be familiar with that. You should also know that smooth muscle lacks troponin. There is no troponin in smooth muscle. And then if you look at the illustration here, so this is a sort of an idealized representation of one of these smooth muscle cells. It's like a kayak to me, but here's a smooth muscle cell. And what we're looking at these filaments, these interior proteinaceous filaments that extend in sort of a crisscross pattern through the cell cytoplasm. Within the smooth muscle cell, there are what are called dense bodies, and dense bodies are simply points of attachment for the filaments. And then at the surface of the cell, the points of attachment are present, but they're not called dense bodies, they're called plaques. So these actin filaments, chiefly actin filaments that you see indicated in this crisscrossing pattern here, extend between intracellular dense bodies and membranous plaques. And when they contract, you get again this globular sort of irregular contraction. As I said, I'm not going to spend any time talking about the biochemistry of contraction. I'm going to summarize that in a couple figures to follow shortly. But another important difference between skeletal muscle and smooth muscle. Remember, in skeletal muscle, the myosin cross-bridge heads of the thick filament. The myosin cross-bridge heads of the thick filament in skeletal muscle were all at the distal ends of the filament. Not so in smooth muscle. The myosin cross-bridge heads, remember, myosin is much less abundant, but you have many more actin filaments all the way around it in 3 dimensions. The myosin cross-bridge heads run the entire length of the molecule. Filament, I should say, I'm sorry. All right, as I say, I do want you to be familiar with this dichotomous classification of smooth muscle. We talked about single unit and multi-unit. In general, the terms refer to the fact that in single unit smooth muscle, if the smooth muscle is classified as single unit, it more or less contracts as one. Hesitate to use the word syncytium, but it contracts as a single entity. In multi-unit, the fibers have the ability to contract independently. And even in this very schematic illustration, I think you see why that is. Notice that in multi-unit, there is individual innervation of the individual fibers by the autonomic nervous system. Whereas in single unit, the entire thing is basically innervated as a single unit, as one entity. With respect to the location of single unit smooth muscle, chiefly in the GI tract and structures related to the GI tract. Also, the small and medium-sized arteries, not true of the large. It wouldn't be true, for example, of the aorta, but certainly small and medium-sized arteries would have single unit smooth muscle. And the reason that single unit smooth muscle can behave as a single unit is because the muscle cell membranes are riddled with gap junctions. So that when one smooth muscle cell depolarizes, adjacent smooth muscle cells depolarize.
Dr. King?
Yeah.
So what is present in the large elastic arteries instead of smooth muscle then?
Well, in the large arteries, you're going to have actually both. You're going to have multi-unit and single unit. The large arteries are a little bit difficult because large arteries, it's like the interstate highway out there. You have little roads on the side, and they have their own laws and principles. Large arteries are so large that they require their own blood vessels, which in turn are regulated in a different way, and they have a different type of smooth muscle. But what we're saying is the lumen of this is the best way to say it. The lumen of the largest arteries is not innervated by, or is not regulated by single unit smooth muscle. It's much more complex than that because it just requires, those vessels are so large they need their own dedicated blood supplies and support cells and so on. I want to come back to this, so don't worry about the contraction too much right now. But I do want to, over on this point, I want to make sure you see that the sort of anatomy of innervation. So we've talked about the autonomic threads or the autonomic fibers before, remember the varicosities. You can see the receptors, neurotransmitter receptors. But notice where the receptors are located. They're only located on cells that are facing or opposing the autonomic fiber. There are no receptors down here. This single unit, smooth muscle is contracting as a single unit because once this cell depolarizes in response to the G protein cascade, and you know why there's going to be a G protein cascade, we fought that battle before. This cell depolarizes, and then what is this? Yeah, gap junctions. And so that there's going to be a rapid propagation. That's a very important word that we use so many times. Rapid propagation of the depolarization into adjacent cells. So that's very different than the way that multi-unit operates and is innovative. So with multi-unit, here I think the locus of the location of the tissues is pretty self-evident. The eye, for example, would utilize multi-unit smooth muscle because of the additional regulatory control required. Portions of the male and female reproductive tracts utilize multi-unit smooth muscle. The fibers, as we've indicated, are not extensively, not noticeably innervated by gap junctions. Rather, if you look at our illustration here, what you'll notice, and they're showing us the ciliary muscle of the eye there, is the autonomic fibers interdigitate between the smooth muscle cells such that there will be receptors for the neurotransmitter on all of these cells, and they will have the ability to respond to the varying concentration of neurotransmitter individually. Again, affording somewhat greater local control and regulation. Yeah.
Excuse me, so do multi-unit smooth muscle fibers generally not have gap junctions?
No, correct. They do not have gap junctions. It's a little bit hard to be absolutely definitive. For example, in the eye, there are sparse gap junctions, but for our purposes, I think it's better just to classify these as having the gap junctions or not. As I say, I don't want to talk too much about the physiology right now. I'm going to summarize that in a couple of graphics. But this just simply shows you what we've already talked about. You can see, for example, here's our thick filament, and in the smooth muscle, you can see the array of cross-bridge heads aligned along the entire length of the molecule. Again, a much higher concentration of actin filaments. You typically in a skeletal muscle may have on the order of dozens of these. In smooth muscle, you have hundreds of these actin filaments surrounding the myosin. All right, so let's get this. Actually, there's one thing I want. Well, let's go. All right, let's go to this. Let's talk about this a little bit. So, smooth muscle has a number of interesting properties that we don't encounter in skeletal muscle. We've indicated that smooth muscle, for example, is capable of the pacemaker potential, which you don't see in skeletal muscle. Another important difference is what was the ionic basis of depolarization of skeletal muscle? That is to say, what is responsible for the depolarization of skeletal muscle? What current? Sodium current. All right. Not true in smooth muscle. Smooth muscle is depolarized by means of calcium currents. Okay. Moreover, I have to be very careful here, but as a general rule, the intracellular calcium concentration determines the force of smooth muscle contraction. The intracellular calcium concentration determines the force of smooth muscle contraction. It's just that in smooth muscle, there are a number of ways that that intracellular contraction can be regulated we won't get into. All right, so in the illustrations here, let's look at the one on the left. You can see calcium entering the cell, and that's going to cause a depolarization. Now, before we get into the details of what follows that, what types of stimuli would induce depolarization of the membrane? That is to say, what would cause this calcium channel to open? Looking for general ideas here. Anybody have any idea what would cause that calcium channel to open and then result in contraction of the muscle? Excellent. You're on exactly the right track. If you go back to the slides, some of those slides I skipped over, well, I want you to understand that there are three fundamental ways that calcium can be induced to enter the smooth muscle cell. Through some type of neurochemical signal, could be a ligand, could be a hormone, could be a neurotransmitter. What else? You remember this drill? You've already been tested on. There's something called intrinsic depolarization of smooth muscle. Yeah, well, remember we talked about stretch receptors. Yeah, stretch receptors can also do it. Smooth muscles can depolarize in response to membrane deformation. That's again sometimes called the intrinsic response. What's the last one? Remember the big three? Voltage gating, right? So all three mechanisms, voltage gating, ligand gating, and stretch, can all induce smooth muscle contraction. All right. So if we come and look at our little simplified representation of smooth muscle physiology here, we can see that, and this is something we talked about last time too, when we talked about, remember the DHP and the ryanodine, let's say that, ryanodine receptors. Do you remember the mechanism that we use to explain the way in which the DHP induced the ryanodine to open the SR door? What was that called, remember? Well, okay, good. It was a relay. You're exactly right. Folks don't generally remember my analogies. I sure don't. But yeah, we called it calcium-induced release of calcium. You can use any term you like to suggest that idea. But that's what's happening here. Calcium, by virtue of deformation of the membrane, binding of a neurotransmitter or hormone, voltage-gated calcium channel. All right, so we have calcium coming into the cell, and that in turn, as we saw before, through a much more complex mechanism than we get into now, induces release from the intracellular SR. Calcium stores from the intracellular SR. All right, so that's calcium-induced release of calcium. Just a wonderful idea. And remember, there's no troponin in smooth muscle, so instead of Instead of troponin, the calcium binds to something called calmodulin. I'm going to put a little graphic in today explaining this process, but it's just a calcium binding protein, intracellular calcium binding protein. And that calcium calmodulin complex, again, in a much more complex sequence that we have time to get into here now, causes a conformational change. It binds to and causes a conformational change in that neck region of the myosin. Remember, the neck region is the light chain. In fact, this is called the myosin light chain kinase. Myosin light chain kinase in that little neck region there. And so then we have the walk along, basically by virtue of this thing changing conformation. It's all, not only does it require the calmodulin, but of course, as we discovered with skeletal muscle, it requires a metal cofactor and ATP. This is going to be a hydrolysis. The myosin head is still hydrolase. And in effect, we're going to have, again, something similar to that cross bridge walking that we talked about in skeletal muscle, resulting in increased muscle tension. But again, the key difference is calcium current, calcium depolarization, not sodium depolarization. We have a similar sort of calcium-induced release of calcium, but once again, the key thing to remember here is that perhaps even to a greater extent than with skeletal muscle, it's the intracellular calcium concentration, intracytosolic calcium concentration, that really determines the force of contraction through this general sequence of events. So I'm going to skip over all this because this is my attempt to explain what we just talked about. I do want you to think about this. I do want you to think about this notion of a pacemaker potential a little bit, because this is going to come up again later. Right now, I just want you to be familiar with the notion. But in the top graphic here, this is a graph of membrane potential. And what I want you to see is that the smooth muscle cell is constantly changing membrane potential. It depolarizes and hyperpolarizes on its own. Later, we'll talk about some of the membrane channels responsible for this. But that dotted line should look familiar to you. What was that all about in our discussion of the neuron? The threshold potential, what was that all about? Remember, everything below that threshold was called what? A depolarization below the threshold was called what? Graded. Yeah, same idea here. So in smooth muscle, you have, again, sort of a graded oscillation, depolarization, repolarization, depolarization. But if that depolarization at any point, and we're going to find this is true in the cardiac muscle as well, reaches threshold, then that will generate action potentials that are responsible for muscle contraction. The muscle contraction is dependent upon those action potentials. So here's the key concept that I want you to make sure you appreciate. There is an underlying pacemaker potential. You could draw it in here if you wish. It would be like the one up here. You can just imagine an underlying pacemaker potential. And so long as that pacemaker potential, with each of its depolarizations, achieves threshold, there's going to be an action potential. And that in turn will produce the cascade of events that we just talked about before. The calcium-induced release of calcium binding, calmodulin, activation of the myosin light chain kinase, and so on. And so in general, this should be very regular and rhythmic. And the rate is going to be affected by a variety of factors. We won't get into that now. But this is the basic mechanism by which smooth muscle has the ability to contract myogenically. What do we mean by myogenically? On its own, right? You saw the Mel Gibson movie when they took the hearts out of the people that were being sacrificed. The heart would continue to beat, right? Same thing with smooth muscle. So this is the point we were making before. The contraction can be initiated by some type of neural signal, voltage-gated, some type of endocrine signal, such as a ligand-gated, it may be a hormone binding to a receptor, or by deformation, stretch-gated channels. So somebody explain this to me. what are we looking at in this illustration? What is this thing?
Ligand.
Not a G protein.
Ligand
Yeah,
gated.
it's a G protein receptor. All right, so it's a G protein receptor, and we have the hormone could be, I don't know, we're going to find a number of hormones do this, but it could be a hormone that is circulating liberally in the blood. It binds to the G protein receptor, and then what happens according to our illustration? What is this down here? That's the G protein. All right, when the hormone binds to the hormone receptor, there's going to be a change in the conformation, the shape of the G protein receptor. The G protein receptor then, when it changes shape, is going to cause dissociation of the trimeric. The G proteins have three, we're going to talk about this more hopefully in the second hour. The G proteins have three subunits. And it's not always the alpha. In this case, they're showing the alpha subunit hopping over here. And so what's this green thing? What's it doing? Well, it's not exactly ligand gated. When this alpha subunit comes over here and binds to this thing, I just sort of want to know what it's doing. How does that green thing make a living? But when the alpha subunit binds to it, what does it do?
Changes
shape to
conformation.
allow
Calcium.
calcium. And I just wanted you to see that this is a form of ligand gating, but we have a G protein intermediary. So when the hormone binds to the cell membrane, this is one mechanism, one very important mechanism then by which the smooth muscle cell can be induced to contract. Because remember, it all begins with that calcium entering the cell. And then you can see over here, the calcium pours into the cell, binds to a calmodulin. We fought this battle before. The calmodulin then is going to activate the myosin light chain kinase. And once again, we get that cross-bridge cycling. Very similar to what we talked about in the skeletal muscle. But in this case, I just wanted you to see how it originates. It originates through a second messenger signal cascade. Here, it's only just one step. Most of them are many, many steps. Most of them are on the order of 10 to 40 biochemical steps in length. And this would be occurring in your GI tract. This is the way that smooth muscle contraction is regulated in your digestive system, to some extent in the cardiovascular system, the respiratory system, and so on. All right, any questions? That's our short overview of smooth muscle physiology. If not, let's move on to the endocrine system. And believe it or not, although it seems we're behind according to the schedule, we're doing not that far, not that bad off. Nevertheless, I'm going to proceed apace unless you stop me. Not that there should be any reason for you to do that. So let's talk a little bit about the endocrine system. Later, we're going to talk about the distinction between exocrine secretion and endocrine secretion. But in general, endocrine secretion involves release of something called a hormone by cells somewhere in the body. Usually, we think of them as being endocrine glands, but they don't have to be. For example, your brain, while it's releasing neurotransmitter, also releases hormones. Your heart releases hormones. Your reproductive tissues release hormones. Your fat tissue releases hormones. Just about every tissue, even though. Can you name a tissue that you think does not release hormones? Anybody want to give it a go? Something you can quiz each other tonight. Name the tissue that does not release. What about bone? Does bone release hormones? Yeah, they've recently discovered that bone releases at least 2 different hormones. We're not quite sure what they do. So my point in going over that is simply that you should not assume that the only source of hormones in the body are endocrine glands. Really, they can come from a variety of sources. And what they have in common, at least in the classical view, is that these hormones are released into the surrounding ECF and wind up in the blood. And remember these hormones, if you take nothing else from you today, make sure you understand that hormones are chemical messengers. They don't do anything as a rule. They don't have the ability to do anything other than just to shout and yell at surrounding cells that I'm a hormone. This is my information. This is I've got something to tell you. But they do that from the circulatory system, the cardiovascular system, and these little hormones that are released into the blood shout to the surrounding cells through receptors. So the key idea here, and this is something we talked about in the nervous system, is that in the absence of receptors, the endocrine message cannot be heard. There can be no biological response to a hormone unless the cell or tissue has receptors for that hormone.
Can you tell me the difference between paracrine and autocrine?
We're going to get to that right now. I just want to make sure you understand the classical definition of endocrine. And part of our problem is that as time has progressed and our understanding of these systems has progressed, the distinction that you described, the distinction to which you refer, is broken down. So let's talk a little bit about paracrine and autocrine. So here we have a cell that is releasing a hormone. The red thing is intended to represent a hormone. And the yellow thing is a receptor. So what's odd about this? This cell releases something, and it has a receptor for the very thing it's releasing. Can you think of any physiological value to such an arrangement? Do you have are your systems set up this way?
Smooth muscle, like feedback.
How do I know not to speak more loudly than I am or to speak more softly? The feedback, yes. This is a simple feedback circuit that basically tells the cell how much it's releasing. And that would be autocrine, auto implying self. This would be a self-regulatory feedback system, or autocrine secretion or autocrine release. But you'll also see that this cell, the product of this cell, is binding to receptors on adjacent cells. We sometimes use the word neighboring cells. That would be paracrine communication or signaling. So the technical distinction between endocrine and these two is that these are all local. These are very, very local. Endocrine communication implies that the signaling molecule, the hormone, is released into the general circulation, travels to every part of your body. Take, for example, the GI tract. The GI tract is releasing hormones. Your stomach and your guts are releasing hormones into your general circulation to travel to your toes and your nose. But where is the site of, where are the receptors for that hormone? Just a short distance from where they're released. All right, so it's a crazy system. But my point is, what we call that an endocrine signal because it travels throughout the entire bloodstream. But as I was saying to Bob, in many cases, some of these, some of these signals, some of these hormones have both endocrine effects and local effects. So where do you draw the line when they do both? And that's just the complication, the complexity you have to deal with. So let's talk about some of the basic characteristics of hormones. Again, this is rather dramatically simplified. Remember, these hormones are traveling in the blood. They travel systemically. But they can come from anywhere. As I've indicated, they can come from the brain. Oxytocin and ADH come from the brain. Vitamin D comes from the collective activity of a variety of tissues, the skin, the liver, and the kidney. A and P comes from the heart, the atrium of the heart, so we don't normally think of the heart as being an endocrine tissue. Also, you've got lots of stuff in your blood. You just think of all the things in your blood. You've got cells in there. You've got these massive immunoglobulins. You've got all sorts of binding proteins, chylomicrons, LDLs, HDLs. So how do we differentiate hormones from all that morass? Hormones, by definition, are present in the blood in very minute concentrations. Look at the magnitude here. What's the concentration we're talking about here, typically? How would you express that chemically? Yeah, nano to picomolar concentrations, and in some cases, especially with some of these neural hormones, only recently have the concentrations been detectable. You should know, as a general rule, we're able to identify many more hormone receptors than we can identify hormones. It's much easier to define a hormone than it is a receptor, and in part that's because these hormones are released in such minute concentrations. And they also tend to have relatively short half-lives, although we have to elaborate on this. Certain chemical classes have shorter half-lives than others. I've made this point as well too, but it's worth repeating over and over, and I'm sure that I will as we continue. Hormones are chemical messengers that are only capable of generating a biological response if there is a receptor in the tissue for that hormone. There's no receptor, there's no biological response. The receptor determines the response, not the hormone. Let's take a break there, and we'll come back and talk about the transport, hormone transport.